Hey there, you guys. Welcome back. We're still on unit 4.2, evolution and natural selection, but we are moving on to part 2 notes, which is evidence for evolution. So as a quick summary, evolution is the process by which organisms change over time. Now, we're not talking about necessarily individual organisms. We're actually talking about how species change over time. And this was first recorded by Charles Darwin, who took a voyage to the Galapagos Islands in South America and studied the tortoises and the finches there. And he came up with 5 key observations that led to his theory of evolution. And that's where we're at today. Today we're going to talk about some of the evidence for evolution. So without further ado, let's go ahead and get going, shall we? And there we go. So the first type of evidence is based on the fossil record. So the fossil record is, when we've all seen fossils, we've seen dinosaur bones and all that, or fossilized plants. If you haven't, come see me. I got some fossils in my classroom. And what's going on is that we can actually analyze those fossils to figure out roughly how old they are. And by studying fossils and learning, when they come from, it helps us to view a record of life on this planet. So it shows us a history of the different life that once existed. So by looking at fossils from these different time periods, we can also see what life was like, 65 million years ago, 100 million years ago, 120 million years ago, and we can kind of compare the life and see how it changes over time. There's a couple ways we can do this. One, we can kind of look at rock strata, which is the different layers, and we can compare the fossils found in each layer. So now we're saying that the lowest layers of the rock, those are the oldest, which makes sense. And then the topmost layer would be the most recent. So if we look at each layer, we see that there are fossils of this 5-sided organism. And then in the next layer, we see a 5-sided organism, but it's also got these little 5 indentions on it. And then in the next layer, which again is another time period, we see a 5-sided organism with these little 5 indentations and 5 spikes coming out of it. And then in the most recent layer, we see fossils that are of a 5-sided organism with the 5 indentions, 5 spikes, but they're longer and they're not necessarily straight, and a circle mark in the middle. So you can kind of compare that here on the left-hand side, how we can see this organism, this species has changed over time. We are not saying that this initial species is the same one as this final one. We're not even saying these middle ones are all the same species. We're saying that modifications and mutations within this first species led to this next species. Modifications and adaptations and mutations in that one led to this next species, and modifications and adaptations and mutations in this species led to this final species. Another way fossils can help us is by looking at like different stages, kind of similar to what we were looking at with the rock strata. But this time we're not looking at it, we're looking at the fossils themselves. Now, if you just look at this very, very first one over on the left-hand side, and this very, very last one, you might be like, Miss Komar, you are on crack. There is no way those are related. But actually, we do believe they are. Now, this last one, this is a domestic horse leg. So there's where the hoof is, and then the bones going up. This very first one is something called Hyracotherium, which we believe is an ancient ancestor of the modern day horse. It existed somewhere around 55 million years ago. Now let's look at some of the other in-between species we've found fossils of. So if we compare this one to this one, Mesohippus, we can see some similarities. The middle nail there is pretty much the same shape here, it's just larger. And we still have two of the lateral digits, the fingers on the side. And this last one, we don't see it on this guy, but we do see an indentation in the bone, which hints that there used to be something there. All right, well, now let's compare Mesohippus to Parahippus. So again, the middle digit nail is looking very much the same in these two. It's just that it's getting, again, it's bigger. The lateral digits aren't as low. In fact, they're much higher up. You can see that the bones are no longer as prominent on the side, and the digits themselves are rising higher. And now let's compare Parahippus to Pleohippus. That middle digit looks, again, very much the same, just larger and now wider. We don't see these lateral digits on the side, but if you look up here, we see remnants of them. So these are remnants of bones that no longer develop all the way down. So that's actually what's left of the lateral digits. We still have some of the bone that remains, they just don't grow as big. Remember how in this one we saw that there was a top and then it kind of thinned out, or was more so behind it and then curved back out? We're not seeing that bottom portion. It curves to the back and that's it. Now let's look at Pleohippus and Equus, the modern day horse. Again, the middle digit looks very, very similar. The nail portion got wider and broader, but apart from that, the overall structure is very similar. And even here, we still have those remnants of the lateral digits on the top. So by looking from one to the next, to the next, to the next, we can see that incremental change, which helps to support the evolution of the horse. And we're not saying that this guy turned into this guy. We're saying that there was some kind of mutation that led to a slight difference in some of the traits, and that allowed these intermediates to survive. So they lived longer, had more babies, soon we had more of these intermediates, and so on and so on and so on until many, 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 many millions of years, 55 million years, give or take, and many changes and modifications and mutations, that little tiny changes that add up to big changes. You now have the modern horse. All right, let's take a look at our second evidence for evolution, that is homologous body structures. So homologous body structures are structures that maybe have slightly different forms in different species or different uses in different species, but they actually develop in all the same way. And what we mean by develop, when after conception, you have the egg and the sperm, they come together, and then you start building the tissues and the organs and all that to make baby. Well, humans aren't the only ones that develop that way. And what we've noticed is that we see a lot of similarities in how different organisms develop. How our arm, for instance, the bones in our arm, the bones in the flipper of a dolphin, the bones in the wing of a bat or the leg of a cat, how they're developed in the same way. Now, yes, they look different. They have different uses, which we'll talk about in a little bit, but that doesn't change the fact that during fetal development, they start forming from the exact same tissue. So the tissue that would form the bones of my arm, there's an analogous version of that in the cat, and that also forms the bones of their forelimb, their front leg. So if you look at this, we have human on the left, cat, whale, bat. Now, yes, these are all mammals, because mammals are more closely related to each other than we are to, let's say, reptiles. But if you look at the human here in the top, your forearm, or not forearm, sorry, the bicep tricep area, we have the humerus, and then we have the radius here and the ulna here. You can see those in orange and tan. Then we have the carpals, metacarpals, and the phalanges. Well, how those develop in us are also the same as how these develop in the cat. So they have a humerus. There's their radius and their ulna. You can see how it kind of sticks up behind, just like it does a little bit in us. They have carpals, metacarpals, and yeah, they have phalanges. Notice that sometimes cats and dogs have that little tiny claw that's higher up, the dew claw. That's essentially a remnant of a thumb, or yeah. Even in whales, their bones are much smaller in terms of comparative to how our bones are. Now, yeah, whales are bigger than us, but if you look at the human, the biggest bone in this is the humerus, as opposed to the whale. The humerus is actually pretty tiny, but they still have a humerus. They have the radius and the ulna, the carpals, the metacarpals, and then they have these long phalanges that stick into the flippers. So they may not be able to use them like independent fingers, but they're still in there and they still help navigate and tilt and move the flipper around. Just like the cat, it's not going to be able to use its phalanges to pick stuff up like I can, but they use them more so for walking. And then we have the bats, much thinner. We have the humerus, the radius, and the ulna, little bit of carpals, metacarpals, and again, long phalanges. So they stretch more so outwards so you can get more skin in between them, and this allows for the wing pattern. So the bones don't necessarily look identical to each other. The function of them is not the same. Humans, we use our arms for pushing, grabbing, carrying, things like that. Cats use them for walking, swatting, maybe, clawing. Whales, we're going to use them to help steer when they are swimming. And bats are going to use them to fly. So very different functions. However, the humerus in all of these organisms develop from the same tissue during the same time in fetal development. Same of the radius, they all develop from similar tissues, and the ulna and the carpals and metacarpals. So they all develop in similar ways. And that shows us that if our development of these structures is similar to this development of these structures in other organisms, that there has to be some kind of relationship between us. You can even kind of see it in embryonic development. Did you know that when you are in the womb, you actually have a tail and gill slits for a little bit during fetal development? Here we show a fish embryo, reptile embryo, bird embryo, and yeah, that is a human embryo. May not look like a human, but that is still a human embryo. And what we notice, first of all, they all kind of have similar shapes to the eyes, more or less similar shapes in the head, just different sizes. And what we also notice, tail, tail, tail. Yeah, humans, we had a tail at one point. That's why we got that little bit of a tailbone sticking out at the base of our spine. It's essentially a remnant, and why every so often, sometimes a child is born with a tail today. Gill slits, gill slits, gill slits, and yeah, we have gill slits for a little bit. They don't stay very long. We actually develop past them and we get rid of them and develop lungs instead, but you still have them for a period of time. So seeing that in early fetal development, we have these common structures. We don't use gills today. I don't see any gills, do you? No, I don't have a tail either. You're just gonna take my word on that. But because of these similarities and how these organisms develop, that shows us that there has to be some kind of relationship between them, some kind of commonality, a common ancestor at some point. All right, so think about it. Which do you think are most closely related? The bees in the bat or the dolphins in the bat? Now, a lot of times people might say bees and bat because they both fly, they maybe have similar food sources and all that, fruits, flowers, nectar. But actually, it's the dolphin and the bat that are most closely related. Yeah, one flies and the other swims, but we see a lot more commonalities in their development. They have an internal skeleton that develops in the same way. Bees do not have an internal skeleton. They have complex organ systems like digestive and respiratory and circulatory and nervous. Bees' internal systems are nowhere near like that. Let's see. They are warm-blooded. They actually maintain their own body temperature. Bees don't do that. They give birth to live young and they develop mammary glands to provide milk to support the baby. Dolphins do. And yes, bats do. So we see more similarities between these. Those embryonic pictures we just looked at, that's not what a bee looks like at all. So we do see a lot more commonalities in terms of development between the dolphin and the bat. So it's not just about the function of a structure. We also have to look at the origin of it. How did it develop in the first place? There we go. All right. Next one, vestigial structures. So what vestigial structures are is they're organs or body structures, something that really aren't useful anymore. That's not to say that they necessarily hurt us in any way. Generally, they don't necessarily hurt us, but they're actually thought to be evolutionary leftovers, meaning an ancestor of ours maybe had a use for this structure, which is why it's in our DNA, but since we've changed over time, maybe we no longer have a need for it. So let's take a look at some examples of these. First off, the appendix. Well, first of all, I am by no means an expert on the appendix or wisdom teeth or human anatomy. So I'm just going off of things that I've been taught and told as well. Now, the appendix is, I know there's been some recent debate. A recent study came out that said maybe there's another function for it, but until that's been verified, most people believe that the appendix is an evolutionary leftover. It's a vestigial organ. Now, what happens is it's thought that maybe this appendix, this structure, this organ, it's a leftover from way, way back in our evolutionary history when maybe we had more of a herbivore type diet. They tend to see appendixes very large and functional in organisms that eat a lot of cellulose. If you remember, cellulose is found in the cell walls of plants. Now, ours doesn't really do that anymore. It just kind of sits there. Sometimes waste and all that gets trapped in there, and that's why it can become inflamed and you need to have, what is it? Well, they call it appendicitis, and you have to have the appendix removed. You're perfectly healthy without the appendix there. You don't really need it. And that's why it's fine to have it taken out. But it's, like I said, it's thought that it's leftover from when we had a higher cellulose diet. Since we don't really have that anymore, if we don't really use it, it doesn't really hurt us. Most of us are never going to even notice it's there. Most of us are never going to have appendicitis. So we can just, it's no pressure to get rid of it. It's not like we're more likely to die with it. So as a result, it just kind of hangs out as a non-functional leftover. And wisdom teeth. Now this is thought that, again, a common ancestor had a more elongated jawline, so it stuck out a little bit more. I'm not talking about like all the way like that necessarily, but stuck out a little bit more. And the wisdom teeth would have fit into that longer jaw very nicely. However, since our face is more so, flattish, it's not like it sticks out that far. Our jaw doesn't stick out that far anymore. There's really no place for the wisdom teeth, which is why most of the time, if someone has them and they start emerging, a lot of times you'll get them surgically removed just because they can't actually push on the forward teeth. Now, could you survive with them in there? Yeah, but it could be painful until everything gets settled. And a lot of times that's why they just take them out. You don't need them. You have plenty of other teeth in there. If you ever put your finger back there, and you can even see here, you do have space for another tooth there. However, with how it forms in the jaw, it doesn't come straight down. It tends to go more at an angle. Again, hinting that there was a longer bone structure at some point. Something interesting is that today they're seeing more people born without wisdom teeth. So could be a possible piece of evidence for present or modern evolution. Tailbones. We have a tailbone. We do not have a tail. Now, yes, we have one for a little bit during fetal development, but then we kind of slurp it back up and it becomes part of our spine and pelvis. But we still have that residual tail. Now, having that little tiny piece of a tail doesn't really hurt us. It doesn't help us, doesn't hurt us, so it's just kind of a leftover from when our ancestors had tails. And again, further proof that at one point maybe we did, besides seeing a tail during embryonic development, is that every so often people actually are born with tails. Now, they're non-functional tails at this point if you're born with one, because we no longer have the genes to code for, nerve development and functional muscles in there. So it would just kind of hang there, as opposed to like a dog, which can move its tail. The whales. Whales are interesting creatures. It's believed, based on, again, fossil evidence and different things like that, that whales and dolphins in their evolutionary history, their ancestors lived on land, but then went back to the ocean. So they lived on land, but then transitioned back to aquatic life. And one of the pieces of evidence for this is, well, if you've ever seen a whale or a picture of a whale or a movie with a whale, they don't exactly have hind legs. Yet, they have small femurs. Now, femur is the bone in your thigh. So if it's the bone in your thigh, that means it is a leg bone, but they don't have legs. So again, this is an evolutionary leftover. They don't need to have femurs because they do not have back legs anymore, yet they still have a small portion of one that continues to remain. And eyes, specifically blind eyes. Now, in many organisms, it's clear why we have eyes, because it allows us to see things. But there are organisms that live in areas or environments or ecosystems where they cannot see anything. For instance, here on the top left, we have a mole. Moles live underground. You ain't gonna see anything underground. So if you look right here where the mouse is, that's actually where their eye is. They do have vestigial eyes, meaning that they have an eye there, but it's non-functional. Even if you were to open up that skin there, it wouldn't be able to see anything. The ocular nerve is not going to detect anything. Same with the fish at the bottom. It's a type of fish that lives in like water, lakes, or underground lakes and all that. Again, underground, no light. You can see that there is a vestigial eye right there. It is not highly developed at all, which is why you can barely see it. Same with the mole, you can barely see it. But it has an eye, but it's a non-functional eye. Because again, you don't need to see underground So they've kind of lost that ability. They still create the eye, it's still in their DNA, but they no longer create functional eyes. All right. And the last piece of evidence for evolution is DNA evidence. We've gotten some pretty cool technology that has really helped open our eyes. So DNA, as you know, that's your genetic material. So we can use a process called gel electrophoresis to look at and compare sections of DNA. We can also use DNA sequencing technology in order to compare specific DNA sequences. And let's say I was to take my DNA and my sister's DNA. We would have a lot of DNA in common because we are very, very closely related. But then if I take my DNA and my cousin's DNA, we're still going to have some DNA in common because, again, we're still related, but we're not as closely related. So we won't have as much in common. And then if we take my DNA and someone all the way on the other side of the world and compare them, we're still going to have, some DNA in common because we're both humans, but we're not going to have as much. We're not going to have that many common sequences that we're going to look at. Because, again, we're not closely related at all. So they can do this, but with species, and they can compare how much DNA do two species have in common. And if they have a lot of DNA in common, then that means that they have a more recent common ancestor. If they have less in common, then that means that they have a much farther common ancestor. So it's more, way, way, way back in history. So the more DNA sequences that two species have in common, the more recent a common ancestor they have. So in terms of what gel electrophoresis is, you've seen examples of that on crime shows, like when they talk about, let me go back in there. When they talk about DNA testing to find if someone is, okay, we got this sample of DNA at the crime scene and we want to test this person's DNA to see if they're the killer, or is this person the baby daddy, or things like that. It's what they do is they take the DNA and they actually kind of use enzymes to chop it up at particular locations, put it in a gel, and then run it down the gel using electricity, hence gel electrophoresis. And the bigger chunks won't move that far. The smaller chunks are going to move farther down the gel, and you end up seeing band patterns like you do here. So what this is, it's not just looking at relationships and fraternity and all that. We can use this, again, the same way that we were saying, by comparing species. So I have these fish here. So species A, B, C, and D are known species. Maybe trout, salmon, tilapia, something else. I don't know. Pick your favorite fish. And a new species of fish was just recently found. Well, I want to find out what is its most recent common ancestor. What is its most... What other species could it be related to? There we go. So what does it have a common ancestor with? Now you see here with the new species, we have a line there, 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 and there. What we want to do is we want to compare those lines to the other fish. So it's got this first one in common with C and D. And it's got this one in common with A, C, and D. This one in common with C. This one in common with B. And this one in common with A, C, and D. So out of the 5 lines, the 5 DNA sections that we see in the new species, 4 of them are exactly the same in the known species C. So that means that this new species is most closely related to whatever species C is. So they're more closely related. They have a lot in common. The next one would be D. So D would be the next closest relative. It has 3 lines in common. then A, which has 2 lines in common, and the one that is least related to, meaning it has a very far common ancestor with it, is B. There's only 1 line in common, so 1 section of DNA in common. Yeah, why are you doing this? All right, there we go. So again, here's one for deer. We want to find out which deer species is most closely related to the common ancestor. So we have the DNA of the common ancestor. We have 3 modern species of deer. We want to figure out which one is most commonly like it. Well, if we look through, okay, this common in all of them. Nope. Common in species 3 and 4. Common in species 3. Nope. Common species 3 and 4. Species 2. Species 3. So species 3 is most closely related to the common ancestor. So if we were talking about how that works, we have the common ancestor, and then we have a split. This would lead to maybe to species 3. And then from here, we would see another split that led to species 2 and 4. All right. And now to our additional resources. So again, have some resources and videos that I would strongly recommend as part of this lesson that you check out, maybe some of them, whichever ones you feel like would help you the best. We have one by me, just talking about the 4 evidence of evolution. Again, things we talked about in this PowerPoint, but I use my board to make it more visual. And then other videos include clearly stated what is the evidence for evolution. So you can see already they're going to talk about vestigial structures. And then, another good one to look into. And then Khan Academy, evidence for evolution. This one, you see they do talk about the fossil evidence as well, among other things. So definitely go ahead, check out some of those. It'll help you understand what the different evidence means. If you have any questions, definitely hit me up in our Google Classroom or through email. And I'll put the link for this Prezi in the description of the video so you can easily access the additional resource links. But that's pretty much it for me. So you guys take care of yourself, stay healthy, and just be awesome. All right, you guys? Take care. See you next time.